Hi, I'd like to introduce our recent work on predicting cancer metastasis. Metastasis is the spread of cancer cells to other parts of the body, and it is the major cause of cancer death. And this shows the overall framework of our approach. In this figure, E represents the expression of mRNA genes and M represents DNA methylation beta values. To find changes in correlation between E and M, we first computed Pearson correlation between E and M using N normal samples. And we then selected PCCs below minus 0.3 because DNA methylation is negatively correlated with gene expression. We then added a single tumor sample to the N normal samples and recomputed PCCs using N plus 1 samples, and delta PCC was obtained by subtracting PCC in N normal samples from PCC in N plus 1 normal samples. And delta PCC represents the change in the correlation due to the single tumor sample added to the N normal samples. After performing the Mann-Whitney test on delta PCC between the metastasis group and non-metastasis group, we selected delta PCC with p-value below 0.05, and the number of delta PCCs was further reduced by applying lasso regression, and the final delta PCCs were used as main features in predicting metastasis in our study. Since DNA methylation and gene expression observed in lymph node metastasis are different from those in distant metastasis, we built two separate logistic regression models, and we classified tumor samples into three types based on the TNM stage index. So samples with no metastasis, and samples with lymph node metastasis, and samples with distant metastasis. And the logistic regression models were built with these parameter values. And this table shows the number of samples used in predicting two prediction models. And this table shows the number of delta PCCs left after each filtering step. And both DNA methylation datasets and mRNA expression datasets were randomly partitioned into training set and test set in the ratio of 6:4. And due to the randomness of the partition, we evaluated the performance by repeating 10 times. So all the performance results shown in this poster are the average of 10 runs. And for comparative purposes, we tried 3 different combinations of the features, which are DNA methylation beta values and delta PCC between E and M, and delta PCC plus N values. And as you can see here, all the 3 combinations of features showed relatively high performance in all cancer types in predicting lymph node metastasis and in predicting distant metastasis as well. But the combination of delta PCC with N values achieved the best performance on average. We also compared the features in our study with other features, and so the delta PCC combined with N values are the features used in this study, and delta PCC between microRNA and RNA are the features used in our previous study. And this column shows the performance of expression values of microRNAs and RNAs involved in this feature. And the last column shows the performance of the expression of the 191 genes which are found by other study as the metastasis predictive gene. So as you can see here, delta PCC combined with methylation values shows the best performance. And it is interesting to note that expression values of microRNA RNA or the expression of the 191 metastasis predictive value showed very low performance. In summary, we constructed logistic regression models for predicting lymph node metastasis and distant metastasis, and testing the models on independent datasets shows that the differential correlation between DNA methylation and gene expressions are very powerful features in predicting metastasis. So our method and the features found in our study will be useful for predicting cancer metastasis. Thank you for your attention.